And Eesha is another example of like a perfect matching between the dreams and the project, because I remember in her application she wrote that her dream is to be a biomedical engineer and to build devices to help improve the health condition in third world country. And as you will see during her internship, she learned how to build some kind of devices.
Hello, good morning. So for the past 6 weeks, I've had the pleasure of working in the Yasuda lab. And during the 6 weeks, I worked on a project entitled The Assessment of a New Electroporation Device for Organotypic Hippocampal Slices. So you may be wondering why I'm doing this project in the first place. Well, in the Yasuda lab, otherwise known as the Neural Signal Transduction lab, they study signal transduction pathways of specific proteins that underlie learning and memory. So in order to do this, they have developed sensors linked to fluorescent proteins that study the role of them in the spines of a neuron. As this image depicts, this is a sensor developed by the Yasuda lab, CAMK2 alpha, and this just shows the lifetime of the spine throughout. A spine is essentially how the neuron communicates. So in order to accomplish their goals, they need to transfect their cells. So transfection is a method of delivering DNA into the cells as they will express their foreign DNA. So there are 4 main types of transfection. One being lipofection, which is mixing the genetic material with liposomes so they form a vesicle and permeate the cell membrane. Another being viral methods, which is using a viral vector to infect the cells so they will express the foreign material. Another being biolistics, which is very commonly used in the Yasuda lab. This has gold particles, which are coated with DNA and are actually shot into the slices so they'll express the foreign DNA. And lastly, there is electroporation, which I will be mainly discussing with you today. So electroporation. This is using an electric field to transfect polar molecules into the cell. So as this image depicts, the green being the DNA, the positive being the positive charge, and the negative being the negative charge, a cell membrane is placed onto an electrical field, and a current is passed through, inducing temporary pores. Now DNA is polar, meaning it is negative, so it will travel from the negative end to the positive end through the cell membrane. Now, some of this genetic material will get trapped inside of the cell, and when the temporary pores close, the gene then can be expressed. So there are numerous advantages to using this type of transfection, one being it is fast, it is highly efficient, and it is easy to perform. So currently there is a cuvette system which does use electroporation for transfection. So in this system, they have to either cut the cells and electroporate and then place them back on a membrane, or use cells in suspension. So my mentor had the idea of designing a machine that could allow brain slices on a whole membrane to be electroporated. So this is the prototype, as you can see. There is a top electrode and a bottom electrode, and the chamber can insert right here. And this allows for the current to pass through. So the chambers entail a membrane where the slices can be placed and the foreign genetic material can be placed on top. There are 2 electrodes on the top and the bottom, and that allows the current to pass through. This was made in close collaboration with the machine shop. So when I first arrived here, the machine was already built, and my project was to systematically test this machine and find the optimal conditions for transfection. So this includes a more efficient means than the bialistic transfection method, which is commonly used in the Yasuda lab. This means more cost efficient and more neurons labeled. This also entails sparse labeling. This means finding the optimal amount of neurons labeled to be later used in an experiment. And lastly, to reduce the amount of slices used. In the bialistic transfection method, there are many slices that have no neurons labeled, rendering it useless. This is a waste of time, money, and resources, and this device is increasing the amount of usable slices. So there were a variety of factors and methods I had to test for this machine. Ones that were kept constant are the hippocampal slice culture conditions, and the ones that change are the distance between electrodes, between the top electrode and the bottom electrode, the concentration of DNA, meaning we wanted to find an optimal amount of DNA to transfect the cell, but not too much as it would not be cost efficient. Electroparator parameters, which entails voltages, pulses, and currency. Solutions. In order for a current to pass through the membrane, there needs to be a solution to help facilitate that. So we tested a variety of solutions to help facilitate that current. And lastly, the type of DNA. So mainly I used green fluorescent protein, which is a simple protein used to transfect cells. So these are my results. Here you are looking at an organotypic hippocampal slice. Organotypic means cultivating these slices over a period of time. When the cells are alive, we are able to insert genes into the cell and we can monitor them. On your right is a picture of a much higher magnification of a pyramidal neuron, which we are looking for. The pyramidal neuron is used by the Eesha lab for their experiments, so this is what we were trying to find. So I don't have the time to tell you, but I tested a variety of parameters throughout my entire internship. These are just the best parameters that worked for my goals. The pouring entails actually opening the pores of the cell, and the transfer entails actually getting the DNA inside. So in order for any good experiment, you need a positive control, and we use the biolistic means. This is a tried and true method, and many scientists in the Eesha lab currently use this method. So we used a total of 25 slices for this means, and we got a total of 8 pyramidal neurons. Now for our device, the electroporation, we tried 2 distances using these parameters in a total of 25 slices and got 45 pyramidal neurons. Now that is a significant increase, but it should be noted that some of these neurons labeled were shallow and superficial, meaning they would not be great to use in an experiment. However, these were very promising results. Now, once we got these results, we had 3 different criteria for determining the quality: the cell morphology, the electrical properties of the cell, and the structural plasticity. So cell morphology is essential in identifying the shape, structure, form, and size of the cell. So as you see on your left, this is a pyramidal neuron found in the CA1 region of the hippocampal slice. I took these images here at Max Planck using the confocal microscope. And as you see here, these are the spines on the dendrite of the pyramidal neuron, which are essential in many experiments conducted by the Eesha lab, as I mentioned on the first slide. We did have multiple cells that were labeled, but they didn't show any spines, meaning they were unhealthy. But on the other hand, we also did have many that were labeled with spines. So after we looked at the cell morphology, we had to look at the electrical properties of the cell. So we performed whole cell current clamp experiments to detect the electrical properties of the transfected neurons. This is simply the idea of recording the voltage across the cells and allowing the detection of action potentials, which is the way neurons communicate. We wanted to see if this firing pattern and other electrical activities are healthy. So on your image to the left, this is a pipette entering the cell, attaching, and we could record the currents. And on your image to the right, this is a graph showing the action potentials that the cell demonstrated. So it should be kept in mind that this experiment was very difficult to conduct, as again, the neurons are very superficial, and we do need to work on getting them labeled in deeper layers. After looking at the electrical properties, we looked at the structural plasticity. So when stimulated, spines undergo a structural change. This means that the volume of the spines will grow when stimulated, as demonstrated by this graph. As you can see here, this is the portion where the spine was not stimulated. The volume is very small compared to the rest. And then this red arrow indicates the portion where the spine was stimulated, causing a great increase in volume. Now, the spine should potentiate, meaning it should be grown for a long time to indicate that it's healthy. The spine essentially remembers this activity and should potentiate for a long time. These images are the actual spines that we simulated. This is before. As you can see, it is a lesser volume. This is right after. This peak indicates that it has a greater volume. And this is later. It's not as large as the right after, but it does have an increased volume sustained. This is just a video showing the growth of the spine during stimulation. So, in conclusion, during these 6 weeks, I determined the best parameters that are ideal for the transfection of GFP. And during this internship, working with the machine shop, we changed the original prototype many times in order to have a functioning machine that could work on a day-to-day basis. And compared to the biolistic transfection method, the results were very positive. And it also provided a lower price, as the electroporation was a much significantly lower price than the biolistic method. So as this project is still ongoing, there are future considerations that need to be taken in. One being the metals used to electroporate. Currently, we have tried aluminum and copper, but in the future, we want a higher quality metal to be sustainable and durable throughout. This method was very time consuming, so we are looking to reduce the time it takes to transfect these cells. And lastly, we do need to conduct more trials with more difficult DNA, as the Eesha lab does these experiments on, and that's just some things we need to keep on improving. So I would like to thank my mentor, Dr. Paula Parabueno, for guiding me throughout and teaching me so much that I would never have learned before. The Eesha lab, Dr. Eesha, for allowing me to be in this lab and having this experiment, and everyone inside who I interned, shadowed, or taught me something. I would like to thank the machine shop for working very closely with us, Dion and Marcus. I would like to thank Ilaria for helping me and my fellow interns throughout this entire internship. We would have been so lost without her. And the Brain Trust donors for funding this incredible experience. Thank you so much.
Questions for Eesha? Yes.
So the question was about the metal we are currently using for the electrodes and any future considerations. So currently we have used aluminum and copper, but in the future we are looking at platinum to replace the electrodes.
Other questions? You had a question. I saw you.
So the question was, are we planning on patenting this device? So my mentor is planning on publishing this, and you can discuss this further with her at lunch if you'd like.
Actually, the Yasuda lab has a call open if you have a good idea to name this device. So if you can come up with a nice name, just let us know. Any other question? Yes? Yes. Yes. Oh, question, yes.
So the question is the average time it takes to electroporate. So the whole process took about 10 minutes per slice, as we had to UV it, incubate it, and actually run it through the machine. Yes? So the question was, are these live slices? And yes, they are.
Good job.
Thank you.